for being awake for now, and thank you for the invitation. I'm happy to be here. It's a nice organization also. So yes, today I'm going to present a bit about our work on brain iron accumulation in aging. But first, to put it in context, I want to say that we are what we call ourselves the neuroimaging group. Pay attention to the big A here, and we are quite mixed. So we are psychologists. There are engineers. Then some people skilled in statistics, and our main goals is to study the brain correlates of cognitive decline in aging, both normal and pathological, and also to uncover determinants of neurocognitive aging. And we like to use a lot multimodal neuroimaging. And we are quite attracted to novel neuroimaging techniques. So when iron came out, that it was possible to measure iron in the brain, so then we said we have to do it. So what I'm going to show you, it's a summary of 12 years research on brain iron in aging. So first, why study iron? So first of all, iron is the most abundant metal in the body and in the brain. But we have to distinguish between two types of iron. The hem iron, so the one that is in the blood, bound to the hemoglobin that brings oxygen to our organs. So quite important to have iron. And then there is the non-hem iron. So it's the one that is intracellular. And it's the one I'm going to talk about today. And so this intracellular iron is extremely important for many biological mechanisms, such as neurotransmitter synthesis, synaptic plasticity, myelination, cellular respiration, and many other mechanisms. And so this is here an illustration of ferritin, which is the storage protein of iron. So iron binds to ferritin, and then it's in its ferric form, and it's harmless. So when iron is needed somewhere else, so it gets out of the ferritin, it does what it has to do, and then it binds back to ferritin. A problem that can occur, it's when there is dysregulation of the iron metabolism and there is free iron that is left out in its ferrous form. And then it can become toxic. So I draw your attention on this Fenton reaction. So when there is free iron, it reacts with hydrogen peroxide and it forms free radicals. Oh, here, this is the bad guy. So that induces oxidative stress, production of reactive oxygen species, and further leads to neuroinflammation and finally cell death. And all that is part of an iron-induced programmed cell death called ferroptosis. So I don't go into the details, but and that happens in normal and pathological aging, and that leads to brain iron overload and accumulation. Here it's an example of a post-mortem study done in Sweden, actually, where it shows that in the putamen here, for example, so when we are born, we have basically no non-hem iron in our brain cells. But then it accumulates very quickly as we age until it reaches a plateau. And actually, here it's not obvious, but after a certain age, in older age, it goes up again. So in fact, so it's very good to have iron in young age, in healthy conditions. If you have more iron, is good. But in older age, you have even more iron, and it's not good. And so brain iron overload has been observed not only in normal aging, but in basically all neurodegenerative diseases, especially Parkinson's disease, but also Alzheimer's disease and all the others. So you see it's not specific of a condition, but it's present and it harms the brain cells. So how do we measure iron in vivo? So it's a quite recent technique. It's been around now for maybe, well, now it's not that recent, maybe, I don't know, 15 years. When we started, it was quite new, actually. So it's a sequence. It's called the multiecho gradient recorded echo. So you use multiple echo times. And so it can be implemented in whatever MRI scanner. And from it, you get magnitude and phase images. And from them, you can compute two metrics to have a proxy of iron. You can use relaxometry, which uses magnitude images. Or now what is more popular, it's the other method, the quantitative susceptibility mapping, QSM, and that relies on phase data. So for the relaxometry, it's quite straightforward. We just calculate the relaxation rates across the echo times here. And so we fit just this model for each voxel. And what we can see in the end here is that the MRI signal decreases faster in the basal ganglia, so where it's brighter. So this is where there is more iron, so the R2 star value is higher. QSM is a bit more complex, a lot more complex, actually. It calculates actually it estimates the magnetic susceptibility of different elements in the tissue. And the mostly used method is called MEDI, for morphology-enabled dipole inversion. So the dipole model tells us how magnetic sources, such as iron, distort the magnetic field. And then it also uses structural information, so notably from the magnitude images, to have structural morphological information to be able to solve the overall problem. So from phase and magnitude here to QSM here. So I come back to this image and I zoom in a little bit. We can see some differences between relaxometry and QSM. So relaxometry cannot distinguish between the paramagnetic elements, such as ferritin and iron, and the diamagnetic elements, such as myelin or calcium. So you see that in both the basal ganglia, which is filled with iron, but also you can see the white matter here, so the myelin, it takes the same contrast, whitish. But we have absolute values. For quantitative susceptibility mapping, so it's possible to distinguish between paramagnetic elements, so the ones that are, it's brighter, so again here it's mostly in the basal ganglia, versus what is diamagnetic, like myelin, you see that it gets darker. So even though it's quantitative, it's only relative values, and we need some referencing to be able to work with the data. And there is no gold standard what kind of referencing to use. So both R2 star and QSM have been validated as methods to have a proxy of true iron in the tissue. So they have been validated against postmortem data. So R2 star here, it's the relationship between R2 star and the iron concentration in milligram per kilo. And here it's for QSM, for different regions. So you may wonder why is there more iron in basal ganglia? So we don't know. So I say it so you don't ask the question after. So we don't know. But there is also in cortex, and I'm going to show you some results actually, specifically for the frontal cortex. So first studies on aging and brain iron showed that higher levels of iron are related to more atrophy with aging. Here it was a 7-year follow-up study. So those who had at baseline had more iron in the putamen showed a steeper decline in volume in the same region. And they also showed that having more iron in the caudate nucleus was also related to decreasing capacity in working memory after, I think it was a 2-year follow-up in this study. So we wanted to go further, and we wanted to understand the pathways, how it leads to cognitive decline. So we started a few projects. So before this Iron Age project, we had a mini iron. We started small, you know, with because we were the first ones in Sweden to do brain iron, actually, in vivo in aging. So we had to do some tests before. But then things went well, and we started this big Iron Age project. So we recruited 208 healthy individuals at baseline. We tried to cover adulthood. So they were aged from 20 to 79 years old. And then we did a 3-year follow-up. And we collected lots of data. So we had quite a long MRI protocol. We collected structure, iron, metabolites with spectroscopy, structural scans, including DTI, and activation fMRI, both at rest, but also 2 tasks fMRI. And we also, they had an extensive cognitive and motor battery with different domains, memory, executive functions, and so forth. And then we also took blood, from which we did some genetics and also some peripheral parameters for iron, for example, and for other things like cholesterol and glucose. We also took physiological data, so like blood pressure. And the participants also had lots of questionnaires to fill out on lifestyle, such as diet, physical activity, sleep, etc., etc. So with the aim, again, as I said it at the beginning, it's to study the impact of brain iron load and accumulation on brain function and cognition, and then to unveil the determinants of brain iron accumulation. Then we used another data set from our colleague Ali Reza. It's called the Dynamic Project. It has a similar design, but in addition, there was also a PET scan to measure the dopaminergic D1 receptors. So I show you now just a few studies that we conducted. So first, this study was about its cross-sectional here from the baseline data. And here we try to relate brain iron in the striatum, brain activity during working memory task, the N-back task. What you can first see here, so you can see in the figure that if you observe well, that with older age, it becomes brighter and brighter, so more iron, and here it's a plot for every individual, how it goes. So we see, like the postmortem study that I showed you at first, that there is first quick accumulation of iron from 20 to 40 years old, then it stabilizes up to 60 years old, and then it goes up again. Now, so we run a partially squares analysis. That is a multivariate approach to identify patterns, latent variables that connect all these factors all together. And just to be quick here, what we found, so what you see in red in the brain, it's network associated to working memory. And here what it shows, it's frontostriatal activations. And what this result here says is that in middle-aged and older adults, more striatal iron is related to less frontostriatal activation during the working memory task. You see also the opposite pattern in the younger adults, which means here that more iron is related to more activation. Remember that in younger age it's good to have iron. So that's why we see this opposite pattern. So then we thought, well, so how can we explain that? What's the mechanism behind? So it has been shown that iron accumulates the most in the astrocytes, actually. And the astrocytes, we know they are involved in the neurovascular coupling. So here we have our link with the fMRI, the bold signal. Also, the astrocytes can undergo inflammation. But then, what kind of data can we use for to test this hypothesis? Luckily, we had our MR spectroscopy with our metabolites, and we thought we would use myoinositol, which is a metabolite that increases in case of neuroinflammation. So it's quite unspecific, but some say that it's more related to astrocytosis. But I would not bet on that. But we have a measure of neuroinflammation. So first, we showed that in middle-aged and older adults, there was a significant association between higher iron in the striatum and higher myoinositol. So we have our link between iron and inflammation. If you remember the first chemical, the ferroptosis, that it leads to neuroinflammation. And then we did a mediation model. So you remember we have our association of more iron, less brain activation. We also have our link between less brain activation, worse working memory performance. And then when we put in the model myoinositol, which is related to both iron and activation, the association between iron and brain activity drops. So that means that there is a partial mediation of inflammation. That explains the link between iron and activation. But we still have a link between iron and brain activation. So either it's a direct link or it's mediated by something else. And this is where I come to dopamine. That could be the something else, or one of the something else. So dopamine, we studied a lot in our lab, I have to say. So not me particularly, but colleagues. And dopamine is a crucial neurotransmitter for behavior, for motor processes, and the dopaminergic system goes down with aging. Here is an illustration, an example from a member of the group. Here is the drop of binding potential of the D1 receptors in the caudate and putamen as a function of age. So just a very global view on the dopaminergic system. So here is the dopamine, and then there are two big classes of receptors. We have the D1 receptors and the D2 receptors. And then, interestingly, there is the COMT. It's an enzyme that degrades the dopamine. So in iron age, we don't have a PET scan, but we have genes. And we have the COMT polymorphism. So this genetic polymorphism is very interesting because it can be used as a proxy of dopamine availability. So we have three categories of people. We have those who are Val-Val carriers, those who are Met-Met carriers, and the Met-Val carriers. And in fact, the Val-Val carriers have a higher dopaminergic degradation activity compared with the Met-Met, which means that if they degrade dopamine faster or more, then they will have less dopamine availability compared to the Met-Met, who will have more dopamine availability. So we used this information to study the links between dopamine and brain iron accumulation. And these are the results. First, you see that globally, the Val-Val have a higher increase of iron over time. This is longitudinal, so it changes in iron load over time, over 3 years follow-up. And that is both for the striatum and for the dorsolateral prefrontal cortex. But we see that it's only significant in the older individuals. So here it's a split between young and old, so around 50 years old. So here that means that yes, the Val-Val, so those who have presumably less dopamine in the system, they accumulate more iron over time in both the striatum and the dorsolateral prefrontal cortex. So that would mean that having more dopamine could be beneficial because it has anti-inflammatory effects, so it can block iron accumulation. And in relation to cognition and more specifically working memory, so here it's working memory because we have dorsolateral prefrontal cortex and striatum, so that was the most appropriate cognitive function to study. We see that in both the Val-Val, so those who have less dopamine, and the Met-Val, there is an association between higher increase in iron in the dorsolateral prefrontal cortex and steeper decline in working memory. So in these individuals, accumulating more iron has a deleterious effect on working memory over 3 years only of follow-up. Now I move on to the dynamic project. So you remember it has approximately the same design, but we have a PET scan here for the D1 receptors of dopamine. So it's more or less the same associations. So we have iron with QSM, we have D1 receptors, and we have task functional MRI. So it's the NBAC task. We really like the NBAC task. And that's it. And age. So what we found, so here too, we did associations first between, so this is cross-sectional, between iron and D1. And what we found was significant only for the dorsolateral prefrontal cortex. We found that higher iron was related to lower D1 binding potential. That was not significant in the striatum. And then we did again a partial least square analysis. And this is the final result. I don't show you because it's a final graph because it's many columns in all sorts of directions. So this is the final results. We found that older age, higher iron in the frontal cortex, and lower D1 were associated with lower brain activity during the most difficult working memory task, the 3-back condition of the NBAC task. And moreover, so we always try to relate brain activity to cognition, otherwise what's the outcome? It's lower activity in this the working memory network, frontoparietal, was associated with worse working memory performance for the three-back condition. So I don't know if I have time. Yes. So these were the links. So I just showed you a couple of examples. So we also showed such associations with episodic memory. We also have with resting state, with networks actually, associations and motor function. But these were just a few examples with working memory. Now the question is there something we can do about it? Are there factors that influence how much iron one will accumulate in the brain? So there are two big categories of factors that we can investigate. The genetic factors that are not really modifiable for now, and modifiable factors like lifestyle factors. So we like to focus a lot on these ones because they are important for prevention. Because it's something you can change. So if you can prevent, you can slow down iron accumulation and prevent neurodegeneration. So I start with genetic factors. So there is a group of genes that is called the iron genes. Here I give you the example of one that is well known and that can lead to some serious conditions if you are homozygote. So it's the HFE gene with these two mutations, so the H63D and the C282Y. So this one is when you are homozygous for this mutation, you have the disease called hereditary hemochromatosis. But so we had conditions for the inclusion of our participants in the study, and one condition was that they didn't have hemochromatosis. So we ended up with two groups. We still have individuals with like heterozygous for this HFE polymorphism, and then we had a few more for the H63D. So 66 individuals were classified as HFE positive versus HFE negative, so with no such mutation. So what we found, so it's not huge effects because it's a small sample, it's genetics. So don't expect like huge differences, but they are significant. So first we found that the HFE positive individuals had higher iron load in the blood. So here we used transferrin saturation. Transferrin is the transporter of iron. So when it's saturated, that means there is a lot of iron in the system. So they have more iron in the blood, but they also have more iron specifically in the putamen. So this nucleus is very sensitive to iron accumulation. We often find this nucleus significant. But we found a surprising result. It was in relation to cognition. Here it's just one example with executive function, but it was also significant with episodic memory, for example. And what we found is those with a mutation, with at least one mutation, so the HFE positive individuals, in this group, more iron in the putamen was related to better performance. So here we said, okay, let's take a step back and let's dig the literature. And now comes a little bit of history. So this is what I found in the literature. So the first occurrence and spread of at least this mutation, the C282Y, occurred centuries ago in populations of Northern Europe, including the Vikings. So our participants, basically, or the descendants, which conferred advantages of having increased iron in the body because the environment was challenging, the living conditions were tough. So it was actually good to have more iron for the body. And actually, as a matter of fact, I compared in different populations around the Earth, and there is a slightly higher occurrence of these mutations in these countries of Northern Europe, including Sweden. And our sample is actually representative in terms of percentages. And there have been actually some studies that showed that healthy individuals with HFE mutations, they had higher physical endurance and aerobic capacity. So maybe many athletes have actually have an advantage if they have a mutation like this. But it seems that it also has an impact on cognition, not only on physical strength or endurance. So in fact, most likely if you are heterozygous, these mutations can exert some beneficial effects. So not only for the body, but also for cognition. And we think that it's when iron load has reached a certain threshold that it turns deleterious. Now, let's leave a bit, well, there is still a bit of genetic, but now the big focus is on lifestyle and biological potential determinants of brain iron load and accumulation. So you remember that we had collected a lot of data, and there was very little in the literature with conflicting results and only cross-sectional results. So we used these determinants here. So for blood iron, so we calculated one score where we combined levels of ferritin, transferrin, and transferrin receptors. Then we also created a score of cardiovascular health with the well-known score called the Life Simple 7. It combines BMI, blood pressure, smoking diet, blood glucose, cholesterol, and physical activity. Then we had also a questionnaire on nutrition, and we used this MIND score, so it relates more to like Mediterranean diet. Then we had a different score from the one here for physical activity. We had alcohol consumption, smoking, and a very infamous gene, the ApoE epsilon 4, which is a risk factor for Alzheimer's disease. And we used, as a statistical method, structural equation modeling. I just show you just one example of these dreadful models. I didn't do it. It's Jonathan Gustafsson who did all these models. We went in different steps. So first, we assessed if there was an actual accumulation of iron. So in addition to all the factors you saw, we also used age, sex, education, gray matter volume change, and white matter hyperintensities. Then for each of the determinants, we did one separate model where we used all these variables, also having the baseline and the follow-up to assess changes. And we also did interactions with age and with sex. With age, because we may expect different directions of effects in younger and older people. And in the final model, we took all the significant predictors here, and we put them in the same model because they may be related to each other. For example, physical activity can be related to cardiovascular health. So it was to control for all this. I just show you now the final results. Hello. I don't want to move on. Now. So, one first result, maybe it's not so surprising, is that more iron in the blood is related to more iron in the basal ganglia and more iron accumulation over time. And that was specifically driven by the older adults. So more iron in blood, more iron in the brain. A second main finding in the whole group was that the worsening over 3 years of the cardiovascular health was related to more accumulation of iron in both the cortex and the basal ganglia. Then we had also additional results, but only in younger adults. We found for them that a healthier diet was related to higher iron in the brain, that also more physical activity was related to higher iron in the brain. And here we found a weird result that I don't want to interpret. So only in the younger people, we found that the ApoE epsilon 4 carriers, so risk factor for Alzheimer, was related to more iron accumulation. I would have expected that more in the older people, but maybe we have a selection bias here, I don't know. Or maybe it's something that occurs in young age and then it stays this way. So do I have time for the final study that I want to show? 5 minutes? It's not feasible, but we'll see. Now I show you the last thing that we did with our data, and that is more the work of Farshad, who is over there. I will just describe. So we are interested in other nuclei that accumulate a lot of iron. So for now, I said the basal ganglia, meaning caudate, putamen, globus pallidus, but the midbrain nuclei also accumulate a lot of iron. So it's the red nucleus, the substantia nigra, and the subthalamic nucleus. But it's a bit difficult to access these nuclei because they are small, they are invisible on some MRI images, and moreover, there was not efficient segmentation methods to go capture them and study them. So that's why they are not really studied in the cognitive neuroscience of aging. So I show you how it looks. So on QSM, you can see them pretty well. It's them. They are here and here. And here I put for comparison a T1 image. You see that we cannot see anything. It's like as if they were not there. They are invisible. So here you can see them better if I zoom in. It's just a bit tricky to distinguish between the subthalamic nucleus and the substantia nigra. But when you play with contrast, it helps and you can see the separation. And because I will use FLAIR images as well, I'm showing you that they are actually visible on T2 FLAIR images. So the gold standard is to do manual segmentation. It's the most reliable method, but it needs specialized people, experts. It's time consuming to go and draw one by one all these nuclei. It's a challenging task. It's a decision-making task sometimes. And of course, it's not realistic to apply on large data sets with thousands, hundreds and thousands of individuals. So a solution would be to use deep learning and for an automatic segmentation of this nuclei. So Farshad developed a CNN UNet model. So why? Because, well, it's well adapted for biomedical image segmentation, doesn't need too many training images, and it produces more precise segmentations. So for the training, we used what we call the mini iron data sets with 40 adults. And all structures were manually delineated by me. I did it. And the iron age data set is the one that you know now where we applied it. And in both protocols here, we use T1, FLAIR, and QSM and R2 star, same parameters for the two protocols. So first step was to study the performance of the deep learning model based on which modality we put in. So first we started with single modality, so one by one for QSM, R2 star, FLAIR, and T1, and then multimodal, where we combined 2, 3, and then all the modalities. Then the second step was to select the best input combination and train a model with a complete training cohort, so the 40 subjects, and then we applied it to the big Iron Age data set. And to have a performance metric, we used the DICE coefficient. I'm looking at you now because we talked about it. So Mr. DICE. And so this is just how well it overlaps between the ground truth region of interest. So my hand here was the ground truth, and then the deep learning segmentation. So it goes from 0 to 1. 0, it's no overlap at all. 1 is perfect overlap. So the best model was the combination of QSM and FLAIR. In fact, it wasn't different from the combination of 3 modalities or all the modalities. But why would you load the system with 3 or 4 modalities if just QSM and FLAIR perform as well? So we don't load the system. QSM FLAIR was good. And here is a comparison of this study here so here are the DICE coefficients for red nucleus, left-right hemisphere, substantia nigra, left-right, and subthalamic nucleus, left-right. Here is in comparison with another model that we did a few years ago that was not deep learning, but atlas-based. So it did better. And here it's a previous publication where they also used UNet. Ours performed better, but I don't think that the difference is statistically significant. And then we applied it to the big Iron Age data set, and these are cross-sectional results. So we used both, we fitted both with linear and nonlinear models. And we found that there is a significant increase of iron in the red nucleus, which is nonlinear. So it goes faster at the beginning, and then there is a kind of plateau. But we found linear associations and significant with substantia nigra and subthalamic nucleus. So iron goes up with aging. And longitudinally, so that is the change within the individuals over 3 years, 2.7 years to be exact. It was significant increase over 3 years in the substantia nigra and the subthalamic nucleus, but it was not significant for the red nucleus. So now we continue this work and we combine this with cognitive data to see the relationships of iron accumulation in these nuclei and cognition and motor function. So the summary, I don't think I need to go through this. I mean, I try to demonstrate that brain iron load has an impact. It contributes to brain aging and cognitive aging via associations with dopamine and neuroinflammation. I showed you some genetic polymorphisms that affect brain iron load and accumulation. Iron in the blood and in the brain are correlated. Cardiovascular health can be an important factor to consider to prevent brain iron accumulation in older age. And the usefulness of AI tools to assess this small iron-rich nuclei. There are other iron-rich nuclei that can be investigated. We have 2 ongoing projects on brain iron and neuroinflammation with PET data this time. So data are collected. We are analyzing the data now. And we have another project also with Rodrigo Moreno, Christopher here, with the brain iron in Alzheimer's disease. And I couldn't help, I have to show you. So now we have the 7 Tesla at Karolinska. And it's a project, an upcoming project, where we are going also to collect brain iron data on the scanner, and I'll just show you how beautiful it is. It's the QSM from the 7 Tesla. I was honestly expecting something dreadful because it's the previous ones I have seen from other scanners. It's lots of artifacts, very dirty images. I was really scared, but I mean, we have something beautiful here, so it's very encouraging. And so this is actually the full iron team over the years, over the last 12 years. So it's all thanks to them. And I thank you for your attention.